Hello everyone. Myself Dr. Vivek Kumar, Professor, Himalayan School of Biosciences, Swami Rama Himalayan University, Jorigrant, Dehradun. I am again with you with a new topic on endophytes, that is host plant selection, endophytic bacteria. How the host plant select a particular endophytic bacteria. So endophytic bacteria, they are the plant beneficial bacteria that thrive or present inside the plant, and they can improve plant growth under normal and challenging conditions. They can benefit the plants directly by improving plant nutrient uptake and by modulating plant growth and stress related phytohormones. It means the endophytes, they enhance plant growth, they modulate the host genome, and they modulate the host growth also. So indirectly, these are the direct benefits, and indirectly endophytes or endophytic bacteria, they can improve plant health by targeting pests and pathogens with producing antibiotics, hydrolytic enzymes, nutrient limitations, and priming plant defenses. So we can say that endophytic bacteria, they are generally considered as a subclass of rhizospheric bacteria, so commonly called as plant growth promoting rhizobacteria. So once a bacteria which was PGPR, once it makes an entry inside the host plant, it becomes PGPE, that is plant growth promoting endophytes. So these are in fact a specialized group of rhizobacteria that have acquired the ability to invade the plant host tissues. They share all the important traits consistent with the host plant growth promotion found in the rhizobacteria. It means the endophytes, they enhance, they promote the plant growth in the same way as the rhizospheric bacteria do with their host plant. However, the beneficial effects provided by the endophytic bacteria to the host plants are usually greater than those provided by many rhizospheric bacteria. So such effects may exacerbate when plants are challenged by stress conditions. This stress could be abiotic, this could be biotic. The endophytic bacteria, they have been isolated and characterized from diverse type of plant tissues. These diverse type of plants include your economic crops, your parried plants, plants growing in extreme environments, and wild and perennial plants. So we can say that generally speaking, every type of plant found in the corner of any corner of the world, they are inhabited by the endophytic microorganisms. Now let's study the colonization of plants by endophytic bacteria. So endophytic bacteria, they can be considered a subset of rhizospheric bacteria. So compared to rhizospheric bacteria, endophytic bacteria has an added advantage over rhizospheric growth. So living within the plant tissues allows endophytic bacteria to be in close contact with the plant host. So when they come in contact with the plant host, so they readily exert a direct beneficial effect on the host plant, and they consistently, continuously provide nutrients to the host plant. In fact, the endophytic bacteria represent a class of specialized rhizobacteria. They have acquired the ability to invade host roots after establishing a rhizospheric population. Another benefit of endophytic microorganism is that, so whatever the energy is spent in competition in the rhizosphere, that energy is being spent in promoting the host plant growth, because in rhizosphere there is a lot of competition between the different types of microorganisms. In the endospheric zone, that competition is not observed there. Now this picture shows that this is the root microbiome, you see. So this enhanced root portion shows rhizosphere zone, rhizoplane zone, and root interior zone. So the rhizosphere zone shows the rhizosphere microbiome, and this rhizoplane zone, the rhizoplane microbiome, and this root interior zone shows the endospheric microbiome. So whatever the microorganisms they are present inside the plant, inside the root, they are known as the endophytes. So the endophytic colonization of host by the bacteria is determined by a battery of different bacterial traits. So these traits are collectively referred as colonization traits, and they regulate the entire process of the plant colonization. So this colonization process involves complex communication process between the two partners, that is the microorganisms and the host plant. The process usually starts from the roots and requires recognition of specific compounds in the root exudate by endophytic bacteria. It means whatever the photosynthetes, they are secreted by the host plant, they have certain chemicals, and the certain chemical they will attract a particular group of bacteria. So the plant produce these root exudates to interact with the beneficial bacteria for their own ecological advantage. The host plants select and recruit endophytes through root architecture differences and on the basis of chemical signaling in the root exudates. So the type of root exudate secreted by the plant, that will select a particular group of or specific group of microorganisms. So it has been observed that endophytic bacteria, they colonize the plant interior in a sequence of events similar to rhizosphere colonization of rhizobacteria. So this picture shows that in the bottom you can see the root mucilage is there, that root mucilage that has attracted the microorganisms, and these microorganisms has attracted towards the root mucilage because it contain nutrients, and when they move there they attach to the root surface. If they found any entry, then they make their entry, and slowly we can see this green bacteria, they moves inside the plant and they become endophyte. So endophytic colonization involves a suite of environmental and genetic factors that allow a bacterium to enter plant endosphere. Endospheric bacteria, they usually enter the plants through the root zone, as we discussed before. Aerial parts of the plant, stems, leaves, flower, cotyledons, hydrathodes, they may be used. Once inside the roots, the endophytic bacteria can systematically infect the adjacent plant tissues. Now, colonization of aerial plant tissues by endophytic bacteria. After entering the roots, the endophytic bacteria, they can spread systematically to colonize above ground tissue also. They can establish stem and leaf population with a density between 10 to the power minus 3 to 10 to the power minus 4 under natural conditions. Only a few bacteria, they colonize aerial vegetative parts of the host plant due to the physiological requirements which is needed to occupy these plant niches. So the bacteria that migrate to the above ground plant tissues, they are well adapted to a particular endophytic niche. Let us understand one thing, that some of the bacteria, they are present in the root. It means they are we can say comfortable in that particular niche of the root section. Some of the endophytic bacteria, they have moved from root to the stem, to the leaves, to the other part of the plant. It means they are mobile, maybe some reasons. They have moved with the moving water, with flagella, etc. And then when they move there, when they settle their particular portion, they are comfortable and they have created their own niche. So movement inside plant is supported by flagella and plant transmission system also. The migration along intercellular species is by like enzymes like cellulases and pectinases. Using the enzymes, the microbe, they break the barrier. They break the barrier, and after breaking this barrier, they move ahead. So movement through xylem element occurs through perforated plates and allow movement of bacteria through large pores without requiring cell wall degrading enzymes, and these pores the plasmodesmata. The final sink for this specialized endophytic bacteria is the leaf tissues. They go there and settle there. The endophytic bacteria mostly colonize the leaf tissues from plant roots, but just like phytopathogenic bacteria, the endophytic bacteria, they can gain entry into the leaves from the phyllosphere. Phyllosphere means the leaf zone, from the leaf stomata also. Now, inside a host plant, there is a diversity of the endophytic bacteria. So bacterial endophytes, they have been found in every plant species that have been studied. The endophyte free plant is a rare exception. We can say that rarely there is some plant which is free of endophytes in the natural environment. In fact, a plant without the associated beneficial bacteria would be less fit to deal with phytopathogens, would be less fit to any biotic stress or abiotic stress. So the type of endophytic diversity present in a plant can depend on several factors. So these factors are, we can say, factors affecting diversity. These are the bacterial species, plant specific species or type of cultivar, environments include climate, drought, salinity, season, etc. And then plant health also, and then plant growth promotion is also depends, they play a role in plant growth promotion and the plant colonization. Now, if we discuss this picture in detail, so endophytic bacteria, they can benefit their host both directly by improving nutrient uptake, modulating the growth stage related phytohormones, that is the green box, phytohormone production, plant nutrient provide, phosphate, nitrogen, etc. They manage the stress, drought, salinity, and they also baromediate the heavy metals also. And the grey box, this is the indirect method. antibiosis using lytic enzymes, allelochemicals, induce systemic resistance. The brown box, to confer benefits, the bacteria have the competitively colonize the plant interior like this. When they move inside the plant interior, so they secrete some secondary metabolites because the chemotaxis, etc., they colonize, and they produces, we can say, antibiotics. Because of quorum sensing, they multiply and they prevent the plant. Colonization is a sequential process starting from the plant rhizosphere, followed by the rhizoplane, and from the rhizoplane, the attached rhizoplane, then they move inside. Once inside, the competent endophytes, they can move to the aerial part of the plant. So diversity of endophytic colonizers is affected by various bacteria, plant and environmental related factors. You can see in this sea green box, like climatic factor, environmental specific factors, plant specific factors, like this. There are different factors. Now factors affecting endophytic bacterial diversity of a plant. So apart from bacterial competence to colonize plants as endophytes, the host plant and the other environmental factors, they can strongly influence the endophytic diversity within a particular host plant. So host plant age, its genotype, its geographic location, where it is located in a temperate, semi-temperate region, even the tissues being analyzed, which part of the plant they are present, they can determine the type of endophytic bacteria a host plant harbor. The host plant growth stages can also determine the endophytic diversity stages, means whether the plant is young or 2 years old, 3 years old, etc., because when the plant stages enriched in nutrient availability tend to have increased bacterial diversity. Normally it has been observed that the young plant, they harbor a diversity of microbes as compared to the old plant. Climatic conditions also influence the endophytic colonizers of a plant species. Another important factor affecting the observable endophytic diversity plant is the method of study these bacteria. The spectrum bacteria recovered from the plant can depend on the nature, the concentration of the treatment time of sterilization, the chemicals you have used, the media you have used for the cultivation of microorganisms. Type of endophytic community of a plant is strongly influenced by the nature of plant host species. Different plant species growing in same soil can have distinctly different endophytic community. In one experiment, canola and wheat grown in the same field had very different bacterial species and endophytes, because canola has different genotype, wheat has a different genotype. Obviously they will attract the different types of bacteria. The host species being the most important determinant in selecting its endophytic community, followed by sampling dates, locations. Different cultivars of plant with species grown in the same soil can also differ in the endophytic diversity. So the type of soil used to grow a plant can also determine, say, endophytic community. Same plant cultivar growing in different agricultural soil can have different types of endophytic bacteria. So the plant type, the climate, the soil type, they will also determine the, we can say they are the factor in determining the type of the and diversity of the endophytic community. In one report, different endophytic bacterial diversity for a peanut cultivar grown in different fields. The different types of endophytic bacteria by growing one cultivar of tomato in 15 different agricultural soils. So the agricultural soil, the type of agricultural soil, the type of locations, they also determine the type of endophytic community. Difference in endophytic community can also result by the preference imposed by plant host in response to soil and stress conditions. So plants growing in petroleum hydrocarbon contaminated soils, they recruited those endophytic bacteria which had the necessary contaminated, we can say necessary contaminant pollutant degrading genes. The genes encoding for nitroaromatic compound degradation were more prevalent in endophytic strains selected by the plants than within rhizospheric or soil microbial communities. Because plants select those microbes, the plant sense that these type of bacteria will benefit me. So wilt resistant cultivar of oilseed rapes contained a higher reported of endophytic bacteria which are antagonistic to wilt causing verticillium than the susceptible cultivar. So the presence of phytopathogens in plants have been considered an important factor in the restructuring of endophytic bacterial communities. Hence the selection of endophytic bacterial communities is a dynamic process which is tightly controlled by the host plant. Lastly, the key points: the root endophytes reside in the vast number of plant species as a part of root microbiome. Endophyte community structure within the plant is very dynamic and is influenced by biotic abiotic factors, environmental factors, soil factors, the host genotype factors. Soil environment also influence the plant physiology, affecting biochemical interaction with the roots and the small soil microbiome, which ultimately influence the selection of the endophytes. The plants select and recruit potential endophytes through root architecture difference and chemical signaling in the root exudates. So all these factors, they play an important role in selecting endophytes for a particular host plant. Thank you so much.